strand. Here, a double strand of the double helix is served as a template. Coding strand. The coding strand, otherwise known as a sense strand, is the one that is not copied. It consists of the same nucleotide as that of mRNA, except that it has U instead of T. Transcript factor. A number of potential protein factors are involved in starting transcription. Some of these proteins help in controlling how often genes are transcribed. RNA polymers. In eukaryotes, each RNA polymerase has a specific function. Once the RNA polymerase complex has been bound to the DNA molecule, it unwinds and opens a section of the double helix. Termination sequence. It is different between prokaryotes and eukaryotes. 5-cap. It involves the covalent linkage of a modified G nucleotide to the 5' end of the pre-mRNA. The cap is recognized by the protein synthesis machinery. Poly-A tail. It involves the covalent linkage of a series of A nucleotides to 3' end of pre-mRNA. The tail makes the mRNA more stable and allows it to exit along in the chloroplast. Spliceome. These are a series of small nucleoproteins, SNRNP, that work together to remove introns. SNRNPs recognize specific sequence on the introns. 1, it cuts out the intron sequence. And 2, splices and extron sequence together. Intron, interfering RNA sequences need to be removed before translation. Exons, RNA sequences that will be expressed, helps in making the protein. Transcription unit. This part of the DNA is used to make RNA. Ribosome. These are the areas where translation takes place. It is made of protein and rRNA. Consists of multiple parts known as eukaryotic. It consists of 2 subunits, a large and a small. A, P, and E. Sites. The A site, amino acyl site, binds amino acyl tRNA except from the first tRNA. The P site is the polypeptide site. Amino acid from the tRNA here binds to amino acid on the tRNA in A site. And the E site is the exit site. Here the tRNA exits the ribosome. tRNA transfer. RNA translates between the nucleic acids